Let us now study a very interesting topic, that is of antibiotics. Compounds obtained from some bacteria and fungi for destroying or preventing the growth of harmful microorganisms are called antibiotics. So there are certain bacteria and fungi which secrete certain compounds which in turn can destroy or prevent the growth of other microorganisms. So such compounds are called as antibiotics. Antibiotics were discovered in 20th century, mostly against bacteria and protozoans. We can classify antibiotics into two groups, that is broad spectrum antibiotics and narrow spectrum antibiotics. Now, broad spectrum antibiotics are active against a wide number of bacterial types, and so they can be used to treat variety of bacterial diseases. Whereas narrow spectrum antibiotics are active against only a selected group of bacterial types. So as the name suggests, broad spectrum, they can be used for diseases that are caused by a variety of bacteria, whereas narrow spectrum antibiotics are active only against a selected group of bacterial types. Now let us have some interesting information on antibiotics. We all are aware that the first antibiotic was discovered by Alexander Fleming, and it was penicillin. Alexander Fleming was a little disorderly in his work and accidentally discovered penicillin. So once he had gone for a holiday, and then when he returned back to his lab, he found that there was a fungus by the name Penicillium notatum that had contaminated, or we can say it had grown in a plate of Staphylococcus bacteria, which he had accidentally left uncovered. So when Fleming had gone for a holiday, he must have been working with Staphylococcus bacteria, and when he left, he had kept the plate uncovered, and so the fungus Penicillium notatum had contaminated the culture plate of Staphylococcus bacteria. That is, the fungus had started growing into the plate of the bacteria. Now the fungus had created bacteria-free zones wherever it grew on the plate. So what did the Penicillium notatum do? It started creating bacteria-free zones, means it had started destroying the colonies of bacteria. And wherever it was growing, it was destroying the bacterial colonies. Fleming isolated and grew the mold, that is fungus, in pure culture. So to ensure that what he was going to derive at, what did he do? He started the experiment all over again, and he isolated and grew the mold, that is the fungus, in pure culture. So what did he do? He took a nutrient medium, he added a small quantity of bacteria, that is Staphylococcus, and the fungus Penicillium notatum, and he observed again. And he found that Penicillium notatum proved extremely effective even at very low concentrations, preventing Staphylococcus growth even when diluted 800 times. So when he conducted his experiment for the second time, he had taken very small quantity of Penicillium notatum, but he found that it was extremely effective and it could destroy the Staphylococcus colonies. Then there was a fire that broke out in Boston, and 500 people died. Many survivors were there and they needed skin grafting. But when skin grafting is done, it is liable to infection by the Staphylococcus bacteria. So the treatment with penicillium or penicillin was hugely successful. And so the government then began supporting the mass production of this drug. And by 1944, penicillin was being widely used to treat troops, that is soldiers, for infections both in the field and in the hospitals. And by the end of the World War II, penicillin was nicknamed, that is, the wonder drug, and it had saved many lives. So this is what is the story behind the discovery of penicillin, that is the first antibiotic. And Sir Alexander Fleming shared a Nobel Prize in 1945 along with two other scientists for his discovery. Let us see the definition of antibiotics. An antibiotic is a type of antimicrobial substance active against bacterial infections. So an antibiotic is used specifically only to treat bacterial infections. Hence, it is a type of antimicrobial substance. Now, bacteria are not the only microbes that can be harmful to us. We know that diseases are caused even due to fungi and viruses. So, those which target funguses are called antifungals, and those which target viruses are called antivirals. So, we can say that the term antimicrobial is an umbrella term for anything that inhibits or kills microbial cells, including bacteria, viruses, and funguses. So, an antibiotic is a type of antimicrobial substance active against bacterial infections. Antibiotic medications are widely used in the treatment and prevention of infections. They may either kill or inhibit the growth of bacteria. So any substance that inhibits, that is stops the growth and replication, that is multiplication or increasing in number of a bacterium, or kills it outright, can be called as an antibiotic. So antibiotics are a type of antimicrobials that are designed to target only bacterial infections that are within the body or even on the body. Now, how does a antibiotic work? So antibiotics are used to treat bacterial infections. Now some are highly specialized and are only effective against certain bacteria, which we have seen that they are called as narrow spectrum antibiotics. And others that are broad spectrum antibiotics attack a very wide range of bacteria, including the ones that are beneficial to us. Now, we can recollect that we had talked about probiotics and their uses and applications. And we had learned that when we consume antibiotics, even the good bacteria that are inside our body get inactivated due to antibiotics. And so we should consume probiotics to make these good bacteria active again. So now here we come across this, that is the broad spectrum antibiotics attack a wide range of bacteria, including the ones that are beneficial to us. And so it's important to consume probiotics. So we have seen that how the antibiotics work. That is, we have got two types, that is narrow spectrum and those which are broad spectrum. The introduction of antibiotics revolutionized the way the infectious diseases were treated. And between 1945 and 1972, the average human life expectancy jumped by 8 years, with antibiotics used to treat infections that initially killed the patients. So earlier, when there were no antibiotics, the life expectancy was less because the infections used to kill the patients. But with the inventions and discoveries of antibiotics, the life expectancy jumped by 8 years. So today, antibiotics are one of the most common classes of drugs that are used in medicine, and it has made possible even complex surgeries that have become routine all around the world. So this is what is the importance of antibiotics. Now just we will have a look at the various types of antibiotics. Antibiotics like penicillin, cephalosporins, monobactam, bacitracin, erythromycin, gentamicin, neomycin, streptomycin, tetracyclines, vancomycin are used against various types of bacteria. So all these are antibiotics. And rifamycin is effective against tuberculosis. So this is about the various types of antibiotics that are in use. So all these that are given over here in this paragraph, all these antibiotics are broad spectrum that are used for variety of bacterial infections. Whereas rifamycin is a narrow spectrum used against tuberculosis.